This video is going to cover adult derivatives of different embryonic structures. So at 3 weeks gestation, embryo will undergo gastrulation. And by that, it will form a trilaminar embryonic disc, which has 3 layers composed of ectoderm, mesoderm, and endoderm. Now, the surface ectoderm gives rise to epidermis, hair, nails, tooth enamel, lens of the eye, anterior pituitary gland, which is from the Rathke's pouch, and parotid glands. Now, just to remind you that Rathke's pouch is a depression in the roof of the primitive mouth. Then mesoderm gives rise to muscles, connective tissue, bone, cartilage, blood, adrenal cortex. I would like to remind you that it's the adrenal cortex versus adrenal medulla, which is derived from the neural crest cells. Kidney, spleen, ureter, and dura. So again, just to remind you, dura is coming from mesoderm versus pia and arachnoid, which are coming from the neural crest cells. And finally, we have the GI and the respiratory epithelium, lungs, liver, pancreas, thyroids, tonsils, urinary bladders, urethra, tympanic cavity, and all the glands in the GI tract. Now, just to help you with the urinary system and which structure is derived from different embryonic origins, just to help you out with that one, it's easier if you actually draw it. So here is a kidney, bean-shaped kidney with the ureter attached to it. And then ureter goes to the bladder. And then bladder ends up in the urethra. And then another ureter from this side. So the upper structures, which are kidney and the ureter, are derived from the mesoderm, versus the lower structures, which is the bladder and urethra, are derived from the endoderm. And if you like to add another point to it, you could say that spleen is right here, and thus spleen is also derived from the mesoderm. Now, what else I would like to discuss here is the role of notochord in the differentiation of the ectoderm. So here is the notochord, and it will form the nucleus pulposus of the vertebral disc. So if this is one vertebra and this is another one, the vertebral disc is right in the middle, and the center of it is made by the notochord. Now, another function of notochord is that it sends signal to the ectoderm to cause differentiation, and it will cause formation of the neural plate, and which is composed of the neuroectoderm. So here we have neuroectoderm, and then on the side we have the surface ectoderm, which I discussed earlier, what structures arise from that. And then the margins between these two structures will later on form the neural crest cells. Now, this neuroectoderm will invaginate on itself. So let's go through that. It keeps on invaginating until it forms a neural tube. So that's a neural tube that is formed from invagination of the neuroectoderm. And the other structures that were on the margins between neuroectoderm and the surface ectoderm gives rise to the neural crest cells. And I will discuss what structures arise from neural crest cells in a minute. Now, at the cellular levels, neural tube gives rise to 3 different types of cells. It gives rise to oligodendrocytes, which are required for the formation of myelin in the central nervous system. It gives rise to astrocytes, which are required for physical support and the maintenance of the blood-brain barrier. And finally, it gives rise to ependymal cells, which are in the ventricular lining, and it will help to form the cerebrospinal fluid. Another point that I would like to discuss about the neural plates, about the neural tube, is that the posterior aspect of it right here is also called the alar plate, and it forms the sensory neurons, versus the anterior structure right here that is called the basal plate, and it forms the motor neurons. Now, this was all the description at the cellular level. At the tissue level, neural tube will give rise actually to different structures of the brain, and I would like to discuss that with you. So here we have the neural tube that will form the forebrain, midbrain, hindbrain, and the spinal cord. So forebrain divides into telencephalon and diencephalon, midbrain gives rise to mesencephalon, and then hindbrain gives rise to metencephalon and myelencephalon. Now let's go over these structures and the adult structures that arise from each of them. So telencephalon gives rise to the cerebral hemisphere, so it would give rise to this structure right here. And then diencephalon will give rise to all the structures that are in the center of the brain, so right here. So it will include thalamus, hypothalamus, pineal gland, and the posterior pituitary gland. Then we move on to the midbrain, which is derived from mesencephalon, and it would be right here for the midbrain. And finally, we get to the pons and the cerebellum, which are derived from the metencephalon. So pons and the cerebellum are derived from metencephalon. And finally, medulla right in the end would come from myelencephalon. Now you should also know that what ventricular structures, which would be this column right here, are derived from each of these embryonic origins. And it's actually not hard if you know the anatomical position of each of these ventricular structures. Then you would go back and find out that what embryonic structure it has actually arrived from. And I will walk you through that, and it's not that complicated. So first of all, lateral ventricles, if you remember, they are in the cerebral hemispheres. So therefore, lateral ventricles, which are right here, are derived from the telencephalon, versus the third ventricle, which is right here. And so third ventricle is derived from diencephalon. And then the fourth ventricle is in the pons and medulla. So here is the fourth ventricle versus the third that was right here. So fourth ventricle is in the pons and medulla. So they are derived from metencephalon and myelencephalon. And how third and fourth ventricle are connected with each other are through the cerebral aqueduct. And thus cerebral aqueduct, which is inside the midbrain, is derived from mesencephalon. Now I would like to go through the adult derivatives of the neural crest cells, and the memory aid that I use for this is called Motel Pass. So M goes for melanocytes and meninges. Just to remind you, it's pia and arachnoid. Remember that dura is not derived from neural crest cells, but rather from the mesoderm. Then we have odontoblasts, which give rise to the dental pulp, tracheal cartilage, enterochromaffin cells, which are actually found in the GI tract, so they are in the lining of the GI tract, and they help form serotonin. And another name for enterochromaffin cells are actually Kolchitsky cells, so Kolchitsky cells. Then we have the endocardial cushions, which are helping to form the interventricular and interatrial septum, laryngeal cartilage, parafollicular cells, which are found in the thyroid, and what they do is that they secrete calcitonin, and calcitonin decreases the concentration of calcium by inhibiting the absorption in the gut, in the kidneys, and also inhibiting the function of osteoclast. Neural crest cells also give rise to the peripheral nervous system, adrenal medulla. So again, adrenal cortex was deriving from mesoderm versus adrenal medulla, which is coming from neural crest cells. Aorticopulmonary septum. So we have the aortic trunk, and then the septum that is right in between, separating aorta from the pulmonary artery, is actually coming from neural crest cells. And then we have Schwann cells, which help myelinate the nerves in the peripheral nervous system. So it's required for myelination of the PNS. And finally, sensory and autonomic ganglia are derived from neural crest cells. So if you go through this Motel Pass and repeat it a few times for yourself, then you'll get a hang of it. And that concludes our discussion.